Namaste to all. Welcome to the next session on cell biology and genetics. So today's session, we are going to speak about eukaryotic cell division. As we have already seen about the cell phases or the cell cycle, we know that now the cell is already completed its interphase. It is about to enter into the next phase, that is the mitotic phase. In this phase, if you see, there are three different types of eukaryotic cell division which is discussed in this particular topic. The first in this is called as mitosis, which is a direct division. Second is mitosis, which is indirect division or equational division. And third type is our meiosis, which we will deal in the next session, called as reduction division. Mitosis, if you see, as I said, this is a direct division, contains two different parts. So it has karyokinesis and cytokinesis. Karyokinesis, if you see in this process of mitosis, there will not be any division as such, but the nuclear division is going to take place. Without the formation of any spindle fiber, the invagination of the nucleus happens, due to which the cell wall also contracts and undergoes the constriction, due to which the cell and the nucleus, they divide. When they enter into the cytokinesis, this is going to increase the constriction process, and cells like bacteria, mainly the cyanobacteria or yeast, cartilage cells, the cells which are extra embryonic membrane like amnions and karyons, etc., endosperms, disease cells and old cells undergo the process of mitosis. The main drawback of this particular process is, since the nucleus is undergoing the constriction process, the equal division of the nucleus and the cell will not happen. That is, the chance of 50-50 division will not be there. This is an unequal distribution of chromosomes. That is why we call this process as direct division or mitosis. Let's see about the next process in this, that is our mitosis. Mitoc means thread and osis means stages or state. Scientists Sternberg in plant and Fleming in animal observed this process, and Fleming was the person or the scientist who coined the term mitosis. It takes place in haploid and diploid cells, mainly in the somatic cells. It also happens in the germinal cells during the multiplication phase. Also, this is called as equational division, which we will see in the coming topic. So, let's see about the stages of mitosis. It also has two processes. One is karyokinesis, which is the nuclear division. And the second process is cytokinesis, which is the cellular division or the cell division or cytoplasmic division. Karyokinesis consists of four phases: prophase, metaphase, anaphase and telophase. If you see why we are saying this as an equational division is because in this phase, the parent cell, which is having the 2N as a chromosome after the interphase, or the C value is 4C, is now going to convert it into two daughter cells, and during that process, the 2N and the 4C value is being maintained here. That is why now the 4C, what we have said in the parent cell, since it is divided into two daughter cells, it is going to form as 2N and 2C in both the cells. So, let's see about the stages of mitosis. In this part, we have to understand that the animal cell is already present in the interphase now. During this, we can see that near the nuclear membrane, we have our centriole, cell membrane is intact, nuclear membrane is present, chromatin fibers are there, nuclear pores are there, and cytoplasm is intact. In this phase of interphase, we can see that the parent cell chromosome number is 2N and the C value is 4C. So, let's see about the first phase in this, that is our prophase. In prophase, we have two stages. One is early prophase and late prophase. If you see in early prophase, as we have seen the cell, now the components or the organelles which are present here, they undergo the dissociation. The dissociation of nuclear membrane, then it has Golgi apparatus, then it has the smooth and rough endoplasmic reticulum which undergoes dissociation. Due to the dissociation, now we have only the nuclear membrane which is present, which has expanded, we can say here, in this particular phase. The centrioles are present on the top, and now the centrioles are going to move towards the opposite direction. So the two centrioles which are present, they move towards the opposite direction, and when they move towards the opposite direction, the nuclear membrane is completely dissociated. Once the nuclear membrane is completely dissociated, the chromatin fibers which are present here, they undergo condensation process, due to which they form into the chromatin. The chromatin fibers, they become as chromosomes. Now, in the second phase of the prophase, that is our late prophase, we can see that the centrioles releases smaller and larger or bigger fibers. The bigger fibers are called as spindle fibers, and the smaller fibers are called as aster rays. Since two aster rays are present, or two asters are present on each pole, this process is called as amphiastral. So each chromosome, they have two arms, and the DNA is already synthesized or replicated. It is ready now. The fibers which are released from the asters and the bigger fibers which are present, they are going to attach with the chromosome arms. Once this process is completed, now they enter into the next phase, that is our metaphase. So in the metaphase, we can find that the chromosomes which are there, they align in the equatorial plane. Once they have aligned at the equatorial plane, due to the differentiation concentration of the chromosome spindle fibers, the spindle fibers, they attach to the kinetochore of the chromosome. Once they are attached, now they are pulled towards each other, that is on the opposite poles. So the two chromosomes which are present, they are intact like this, and in the sandwich method, they are going to be pulled on opposite direction now. So once this process is completed, we can see that they enter into the most important part of this phase of mitosis, that is called as anaphase. In the process of anaphase, what we can observe here is the chromosomes are in the equatorial plane, spindle fibers are attached, and it is present in the centrioles. From the centrioles, the aster rays are there, as well as the bigger fibers are there. In the bigger fibers, we can find that they are either connected to the chromosome or they are discontinuous. That is why the connected ones are called as continuous fibers, and the ones which are not connected, they are called as discontinuous fibers. So during this process, now as we have seen in the metaphase, that the chromosomes are going to be pulled up from upside and the downside part. Now what is going to happen is, in this phase, we can find that intrazonal fibers are going to be formed. Once the intrazonal fibers are formed, now what we can see here is there are two phases which are present here. The first one is called as early phase, in which the intrazonal fibers which are formed here will be between the chromosome will be shorter, and the chromosome fibers which are coming from the aster or the centrioles are bigger. Later on, when they are pulled apart and they move to the opposite poles, what we can see is the intrazonal fibers are becoming bigger and the chromosomal spindle fibers are become shorter. At this phase, the intrazonal spindle fibers or intrazonal fibers are becoming weaker, and thereby they lose their control and they dissociate from that and they reach to the opposite poles. So in this phase, what we can observe here is, we can observe that the spindle fibers which are there, now they are in the two forms, intrazonal fibers and in the chromosomal fibers. So as we have seen in our previous slide about the animation of anaphase, Now let's see what other things are happening. So centromere, which is splits, and it is creating a fiber called as intrazonal fiber. There are two phases which are happening in this. One is the early anaphase and late anaphase. Early anaphase, as I said, it is going to have intrazonal fiber in a shorter form, and the chromosomal spindle fiber are going to be longer. Late anaphase, we will see that the chromosomes are split and they have moved to the opposite poles, so the intrazonal fibers are larger or bigger and the chromosomal spindle fibers are shorter. So the forces responsible for the movement makes the chromosome from the equatorial plane to the poles, and thereby the contraction of the spindle fiber takes place, due to which now the stretching of the intrazonal fiber has already occurred and it has become weakened. So during this process, they require lots and lots of energy. The energy requirement by the chromosome is approximately 30 ATPs are required. And what we can observe in this space is we can find 4 different types of chromosomal formation. The first type, what we are going to see here, it is called as metacentric. The metacentric means both the arms of the chromosome will be approximately equal in number and they look like a shape of K. The second part in this is your submetacentric. In this submetacentric, one arm looks shorter and one arm looks longer of the chromosome, due to which the shape of L is resembled. The third in this, what we are going to see here, is called as acrocentric. In this, one of the arms becomes shorter and other arms become longer, due to which they take a shape of J alphabet. The last in this is our telocentric. Since the one of the arms which is there, it is hidden or it is not being present, so due to which only one arm is present and it looks like I shape. Now let's go into the last phase of the mitosis, that is our telophase. In the telophase, we can say this as reversal of prophase. Why we call this as reversal of prophase is, as we have seen in the prophase, dissociation of nuclear membrane, then Golgi apparatus and other organelles are taking place. Here in this phase, the reappearance or reassembly of the nuclear membrane and other organelles are going to take place. The chromosome decondenses to form as chromatin fibers. Astal rays which are present and the spindle fibers which are present on both the poles dissociates and disappears. And other membranes starts to appear, and the cytokinesis phase begins here by the invagination of the cellular membrane. So this dissociate or the cytokinesis is basically we can see in the late anaphase or in the telophase. So in the cytokinesis phase, what we can observe here now is we can observe that the invagination which has started in the telophase is increasing inside and making a furrow towards each other. When the furrow increases, we can find that they are getting deeper and deeper. Thereby, once the furrow reaches to the point where they can join, and then the cell will divide. Due to this, the two cells can be observed which are called as daughter cell, and later on they separate, giving us separate nucleus, cytoplasm and other organelles. Now in this particular stage, the chromosome number remains as 2n and the C value remains as 2C. So due to this division, this is called as equational division, because the number of chromosome present in the parent is same as the number of chromosome present in the daughter cell. So let's see the significance of mitosis. Main type of division which is responsible for the growth. It helps in repair and regeneration. All the cells formed by this division, it helps in maintaining the chromosome number. Also, it helps in asexual or vegetative reproduction in lower organism. With this, I hope that you got an idea about mitosis. Thank you. Have a great day.